Continuing here with the plasmid PUC19, one of the earliest vectors that were used. It's a plasmid, and it shows all the features that a plasmid should have to be useful as a cloning vector or as a vector in general. So some of the things to point out here: the ampicillin resistance, that helps you to screen then for those bacteria that have picked up this plasmid, because if this plasmid conveys ampicillin resistance, then you can grow bacteria that have picked it up in agar that contains ampicillin. So it's a very useful screening tool, because then you can eliminate all those bacteria that did not pick up the plasmid. The multiple cloning site here contains 3 cut sites for Hindi3, BAMH1, and EcoR1. That's the newer plasmids all have a lot more different choices here, but it shows you sort of the basics of a multiple cloning site, and then also an origin of replication. The LAG-Z gene here is for blue-white screening. It was an old method that was used decades ago, I guess. But anyway, this is still a good cloning vector, an early one. It shows the sort of the features that a cloning vector should have. Now we're moving on to a very useful tool for biotechnology, and that's called polymerase chain reaction, abbreviated PCR. And think of it as the DNA copy machine. Basically, you need 3 main things. You need a pair of primers, and then you need a polymerase, DNA polymerase, and you need a template DNA, and then of course a PCR machine. But basically what you're doing here is you are going through cycles of separating the double strands of DNA, annealing primers to a known piece of sequence, and then finishing the sequence between the primers with the DNA polymerase. It's probably easier to just take a look at it on a diagram. So let's take a look at it here. The polymerase chain reaction, the first cycle would be the target DNA right here. So we have a piece of double-stranded DNA. We're going to heat it up, and almost to boiling temperature, where the DNA denatures and it becomes 2 single-stranded pieces of DNA. So you see here single-strand pieces of DNA. Now you need to have a pair of primers. So essentially you need to know something about the DNA that you want to copy so that you can design the correct primer pair. The primers, these 2 orange pieces of DNA, and they find their matches. You allow them to find their matches. That's called the annealing step. And depending on how long the primer is, it could be somewhere between 50 to 65-ish degrees, where you can have the proper annealing temperature here for these primers. One on this side and one on that side. So one for each single-stranded piece of DNA. And now, once you have a primer, then DNA polymerase will come in and finish the job. And it will go all the way to the end of this piece and all the way to the end of that piece. Once the DNA polymerase has finished its job, then you have 2 copies of the original DNA. So you've doubled the amount of DNA. It's essentially replication, but basically targeted to a specific piece of DNA. And then you go with the second cycle, you follow these 3 steps again: denaturing, annealing of the primer, and then polymerization of the DNA. Then you have 4 pieces of the same DNA, so 4 copies of identical DNA. If you do 30 cycles like this, then you will end up with 2 to the 30th power, or about 1 billion pieces of identical DNA. And that's a significant amount. That's something that you can visualize on a gel electrophoresis, for example. In general, the protocols for PCR reactions, they're about 35 cycles maybe of the 3 steps of denaturing, annealing, and then polymerization. So I would like you to know this process, the polymerase chain reaction, because it's very important, extremely useful. And when it first became available, everybody was just right away realized how significant this discovery was, or this new technique was. And it's basically you will not find a biotechnology lab that doesn't have at least one of these PCR machines. Okay, moving on to inserting foreign DNA into cells. So how do we get a plasmid, for example? Now you've engineered your plasmid. How do you get it into cells? And well, probably the easiest way is to just say, okay, well, bacteria, they like to pick up foreign DNA anyway. So let's just mix the bacteria with the plasmid that I just constructed and see what happens. And that process is known as transformation. You can enhance the rate of uptake a little bit by bathing your cells in calcium chloride solution to give them positive charges on the outside, and that will attract the negative charge of the DNA. So that will help. Also, the heat shock protocol helps, and so all of that you can just use. You can enhance the success of the transformation a little bit, and it's not hard to do at all. If we were on site, we would have done a transformation experiment. That would have given the bacteria, regular E. coli, would have given them the P-glow plasmid, which contains the information for making green fluorescent protein, and that would have caused our bacterial cells to glow green fluorescent. It's very nice, and looks really cool. Unfortunately, you don't get to do this, but I will see that I can get you some sort of simulation lab that shows you how this is done. Electroporation increases the efficiency, basically you're zapping those cells with electric current that zaps some pores into the cell membranes there, and then you can get your plasmid in a little bit easier. So that's also very popular. So that's electroporation. And then protoplast fusion, you're just removing the cell walls first, but these bacteria, they're not happy when they don't have a cell wall. They're very, very fragile. And to fuse the cells like that, it's kind of hard. It's not very successful. So by far the most popular method of putting DNA into a prokaryotic cell would be transformation, and then electroporation also. Okay, so here they give you the example of the protoplast fusion. Again, you would have to remove the cell walls from these cells. So remove the cell walls. There's the cell wall. Gotta take it off. Now they're basically like deshelled eggs, and you can try to fuse these two protoplasts. That's what that's what cells are called that have lost their cell wall. So here they would be fused together to be one cell, and then hopefully the genetic material will combine as well, and then you have a recombinant cell. If you want to get your DNA in by injecting it, that can be done with a gene gun, and that's known as microinjection. That is seen right here. So here you see somebody using a gene gun. A lot of times people are using tiny gold particles that they're coating with the DNA that they want to get into the cell, and then basically you have DNA coated gold bullets that you're shooting into the cell. But with a gene gun, you really don't have any influence where your DNA ends up, but it's kind of a random process where the DNA then inserts. This image here shows microinjection of foreign DNA into an egg cell. That can be done. Also, this is often done for cloning procedures. Here's another term that you often hear in conjunction with biotechnology. It's called genomic libraries. At least in the older days when the high efficiency sequencing wasn't available, what people would do is they would take whole genomes, which are usually millions, if not billions, of nucleotides long, and randomly cut them into pieces, and then clone all the pieces into cloning vectors randomly, and then start sequencing piece after piece, and then try to put this together. So it's sort of this shotgun approach to sequencing a whole organism, a whole genome. But the idea here is that you are shattering the genome of a species, of a cell, a cluster of cells, and then you are cloning the pieces that you've generated into the cloning vector, and then hopefully get at least one of each piece of DNA. So here's an example for this, how to generate a genomic library. you take the genome of some species that you're interested in and you're shattering it into pieces, and then here, so this shows on this purple gene right here, purple stretch of DNA, you're shattering it into the green piece, the orange piece, and the red piece. And then the idea here is that you have a whole library of plasmids that each contain a different piece from this DNA that you started out with. So here's the plasmid containing the red piece, the orange piece, the green piece. Or you can use a phage library where you're then using a bacterial phage, for example, that contains here the red piece, the orange piece, and the green piece. So there's different ways you can do this. Plasmids, they can't hold a whole lot of DNA. Bacterial phages, they can hold a little bit longer stretches of DNA. Another term that's kind of getting older and it's not so much in use anymore is the concept of the cDNA library. So cDNA libraries, C stands for complementary, or I usually say copy DNA. So basically in eukaryotic organisms, we talked about the issue with the introns. So if you are only interested in coding sequence, then going to a genomic library is kind of a lot of waste, because at least 98% of the eukaryotic genome in most species is introns that don't code for protein. So to get around that, what people have come up with is that you are extracting the messenger RNA from a species, from a group of cells, for example, and then you take an enzyme that's called reverse-transcriptase to go back from messenger RNA back to DNA. So remember the whole concept of the dogma of molecular biology, where we're going from DNA. Let me write this down. The big concept is always the flow of information goes from DNA to messenger RNA to protein. Okay. So now the idea here is that if we extract messenger RNA from the cells and then we're using an enzyme that gets us the more stable DNA, then we're going backwards. So DNA to mRNA is called transcription, and now we're going backwards. That means we're doing reverse transcription. We had retroviruses before, they were doing the reverse transcription. So here you're using this trick, basically using this enzyme reverse transcriptase, which you definitely need to know. And we're going from messenger RNA that we have extracted from cells of interest. And we are then going back to making DNA that we can then clone, for example, to cloning vectors. But DNA is always easier to work with. It's much more stable than RNA. And this way, you know that you're only going to have coding sequence, because messenger RNA, that's what it is. All the introns were cut out by the cells. And now the messenger RNA was ready to be translated to protein, but instead we took the messenger RNA and made DNA by reverse transcription, which only has the coding sequence then contained. So here would be the cDNA library. So here the concept being that in nucleus we have, so of course this would be a eukaryotic cell having a nucleus. Here's our DNA molecule with a bunch of introns here. And it's a lot of introns. So you're making an initial RNA transcript, and that RNA transcript, then we're removing the introns by the process of splicing, and then we're putting all of the coding sequences together to form messenger RNA that then leaves the nucleus and goes into the cytoplasm. In the cytoplasm, then we can use the enzyme reverse transcriptase, and then instead of making protein, we're making this complementary DNA that has only coding sequence, because it was made from the messenger RNA, which is only coding sequence. Here's another useful tool for biotechnology, and that's a DNA synthesis machine, and it does what the name indicates, it makes DNA. So here's one of those machines. On the bottom here you have the nucleotides separately, and then you can program the machine to add nucleotides as you please, as you wish. That's how you also make the primers for your PCR reactions. You just have to give somebody that knows how to operate one of these machines, you give them the sequence of what you want, and next day you're going to have it delivered to your lab and you can do your experiments. Okay, now we're going to move on into the selection process, because none of these procedures have ultra high efficiency, and you will always have those cells that did not pick up the DNA or didn't pick it up the way you wanted it. And so you need to have a screening tool. And one of the earliest screening tools that became available, besides the antibiotic resistance, the ampicillin resistance, that was the first one, was the blue-white screening. And we'll continue that on the next section.